This episode of Cell and Gene, the podcast, is brought to you in partnership with Thermo Fisher Scientific and Applied Biosystems' new QualTrack real-time PCR and digital PCR solutions for biopharma. Give your cell and gene therapy development an edge with reliable and accurate qPCR and dPCR workflows backed by a trusted supplier. Explore the complete ecosystem of CGMP-compliant qPCR and dPCR assays, master mixes, and instruments at thermofisher.com/qPCR/biopharma. Hello and welcome to this episode of Cell and Gene, the podcast. I'm your host, Erin Harris, and my guest for today's episode is Philip Toleikis, Ph.D. He is the president and CEO of Sernova Corp, which is a publicly traded clinical stage regenerative medicine technology company that develops products for the treatment of chronic diseases such as diabetes and hemophilia, which we will get into in just a second. But before we do, Philip, thanks for your time. Thanks for being here.
Thank you.
Good. So I know I just kind of gave a brief overview of Sernova, but we want to hear it from you. Talk to us about Sernova. What do you guys do?
Sure. So Sernova is a clinical stage public company that is developing regenerative medicine therapeutics. So basically, this is around a cell therapy approach for the treatment of really serious diseases such as type 1 diabetes. And the idea here is that as opposed to just trying to mask disease symptoms, what Sernova is entirely focusing on is providing a functional cure for patients, such as people that have type 1 diabetes. So what does a functional cure mean? Basically, what that entails is if you look at people that have type 1 diabetes, they've lost the ability of cells in the pancreas to control blood sugar levels. And so these cells called islets read blood sugar levels and release insulin into the bloodstream. And so what the patients have been doing for the last 100 years is taking insulin injections to try to control those blood sugar levels. And importantly, if you don't control them really carefully, then you get a series of side effects such as heart disease, kidney disease, blindness, amputations, etc. So super high cost to the medical system of type 1 diabetes. So what Sernova is working on is a cell therapy approach whereby we have developed a small medical device that is placed deep under the skin, which forms a highly vascularized organ-like environment. And then we place the therapeutic cells into that device, and then it forms a mini organ to be able to, again, go back and read the blood sugar levels and release insulin into the bloodstream appropriately. So in our clinical trial that we have ongoing at the University of Chicago, we have for the first time shown 3 people with long-term type 1 diabetes who have become insulin independent. In other words, not needing insulin anymore. So this is what we would call a functional cure. But this is, a stage in the development of the product for all people that have type 1 diabetes.
That's amazing. Okay. And that's exactly what I want to talk about next. So for the trial that's focused on insulin-dependent diabetes, talk to us about where we are, what you've learned to date, any of the challenges that you've had to overcome, and maybe what you're able to implement those learnings into future trials. Where are we at with that?
Yeah, so what's really important is that our first trials, and we've been doing this for a number of years now, there are a number of new entries into the world of cell therapy. Sernova's been working on this for over 13 years now. We have a very significant amount of data from the preclinical perspective and also from the clinical perspective. And importantly, what we have done with our first clinical trial is that we have actually worked with human donor islets. So these are the real things that are in your body that control your blood sugar levels. And we've been able to use those to be able to get a good understanding of the device and how it's functioning, how it's able to keep the cells alive and release insulin into the bloodstream appropriately. So what we have learned so far in this trial is about, importantly, just typically in the same way that you learn in drug trials, about what doses of cells that we need and how much room those cells need to be able to function and read blood sugar levels in a normal way, so that we have a really good idea of what it takes to get patients not only to be able to get some glucose control and eliminate what we call severe hypoglycemic events, but also able to get full working functional cells in their body so that they can eliminate the need for the insulin injections. And what's really important about this trial is that I think everyone knows that the next step for Sernova is really a stem cell derived technology that can treat all patients with type 1 diabetes, and that provides an unlimited supply of cells. So we have done a deal with Evotec, who has developed a highly differentiated islet technology from what we would call an ethically derived iPSC stem cell that has the ability to treat all patients with type 1 diabetes. And so our advanced learning from the clinic with the donor islets is really teaching us, when we get into clinical trials within the next year or so, with the Evotec cells, exactly what dosing we need, and we already have significant safety of our device, and we've reached or surpassed a whole lot of hurdles that other companies have not been able to achieve. And one of those, importantly, is that typically when you put a device in the body, what the body typically tries to do is wall off the device, and in the same way that it walls off a splinter. And so one of the big learnings that Sernova has done and overcome and achieved is, how do you develop a device that you can place into the body that the body accepts as being part of it, and that it actually grows into, so that you have a normal organ-like environment within that device for the therapeutic cells. And we have now shown in our patients that we've been able to not have issues with fibrosis, and that we have long-term function of the device. And this has been, in patients that have type 1 diabetes, and this is proven by consistent levels of C-peptide in the bloodstream that we're finding in our patients. And C-peptide is a biomarker for production of insulin produced by the islets or the cells in our device. So, the hurdles that we've achieved are understanding that we do not get the fibrosis that multiple other companies have had happen to them. And the second one is getting a really good understanding about how many islets we need and how those cells need to be distributed within a device so that they can function really well to actually achieve not only improvement in their hypoglycemic on the worry events, but also be able to get to that functional cure that we're working on.
I do actually have one follow-up question. Your patients, are they across all age ranges? Are they pediatric? Are they adult?
Yeah, so in first sets of clinical trials, we want to have a product that's going to be has the ability to treat all people with type 1 diabetes. And so our first clinical trials, though, are treating adults, and these adults have long-term type 1 diabetes. They have to have severe hypoglycemic events. In other words, when they take an insulin injection, they don't have the knowledge that their blood sugar levels are dropping. They can go into a coma. So therefore, these are what we would consider the severe patients. They also have to prove with a glucose tolerance test that they are not able to produce any insulin. So we are showing that they are not producing any C-peptide in the bloodstream, so that when we do get C-peptide in the bloodstream with our technology, that we know that it's coming from the cells in the device itself. So first clinical trials are typically around, a certain range of adults between 20 to 65. It's very, very standard from the clinical trial perspective. And then as things move forward, then the plan would be to be able to, move the technology into other age groups. But ultimately, we want to be able to have a technology, and this is what we would call an immune-protected cell technology that we can place into all patients. But we have to test safety and efficacy and ensure that is sufficient for FDA at this point.
Sure. Makes sense. Okay. Now, at the time that I was researching the company, at the time you were about to enter the clinic with 2 additional programs. So you had an implantable cell therapy for benign thyroid disease, which was resulting from thyroid gland removal, anticipated to enter the clinic early 2023, and an ex vivo lentiviral factor 8 gene therapy for hemophilia A. So bring us up to speed on where we are with that, and kind of talk us through the vision and the desired outcomes for both of those trials.
Sure. So with the thyroid disease application, there are approximately 200,000 people in the United States who are having their thyroid gland completely removed for benign purposes. And so what we have found is that those patients are not sufficiently regulated by the standard, the thyroid hormones that patients are taking on a daily basis. So these patients typically get severe obesity, depression, lethargy, and this kind of thing. You can look at the thyroid gland essentially as like being the engine in your car. If you remove the engine in your car, you're going to be pushing the car forward to get it going. So what we're looking at, and we've done preclinical studies, and this is in peer-reviewed articles that we've published, we've been able to take the remaining healthy part of the human thyroid and place it into the device in preclinical models. And we've shown long-term survival of the thyroid tissue, as well as precursor thyroid hormones getting into the bloodstream. And it's really a feedback loop between the thyroid gland and the brain that controls the amount of energy that your body has. And this is what we're trying to bring back to the patients. So our first foray into the clinic will be using doing this process where we're able to retrieve the healthy tissue remaining from the patient and place that directly into the pre-implanted cell pouch device. But as we're moving forward, the anticipation is to move forward to an iPSC stem cell-derived technology whereby we could expand our therapeutic treatment to patients that have had their thyroid glands removed a long time ago and don't have any thyroid tissue to replace. So it's really a similar model to what we're doing with type 1 diabetes in the sense where we're testing the real thing, make sure that works, and then we develop like a stem cell derived technology to be able to significantly expand the patient population.
Good. Okay. And that's actually where I want to go next, to talk specifically about the stem cells that, Sernova is using. So explain the type of stem cells you're using more broadly, in the regenerative medicine therapies that you're developing, and why.
Yeah. So with our agreement with Evotec, we actually have had a long-term collaboration with Evotec, where we were working on testing out their stem cell technology in our device in the gold standard preclinical models. And the cells work beautifully. We got long-term insulin independence in these, gold standard animal models. And what was really important to us, and when we first started our clinical trials, were not developed enough, are working with iPSC cells. So these are cells that can be derived from the skin or from a blood sample. And then, what I would call, essentially not embryonic stem cells. These are cells that you can, take from the body that are easy to get, and then you can make a cell bank and then go from there. So we wanted to work with these iPSC cells. And Evotec was the first group, we believe, in the world to be able to get what we would call fully differentiated islet type product that is focused on having the islet clusters be very, very similar essentially to human islets, whereby you've got all of the cell types in there and that they are releasing the right amount of insulin, the right amount of glucagon and somatostatin to be able to fully control the hypoglycemic events and type 1 diabetes, essentially. The other important part of this is that people don't typically talk about is the manufacturing process. So when you're manufacturing a stem cell derived technology, we want to manufacture it similar to a drug, where we have a relatively inexpensive procedure that there is testing at every step of the process to make sure that the cells meet the release criteria and that the product that comes out is exactly the same every single time for every single patient, and that it is cost effective. And this is a commercially developable product that can treat every single patient with type 1 diabetes and bought around the world. And so that's what we found that Evotec has, and one of the major reasons why we did a deal with that particular technology in that company. And Evotec is really important because they have approximately 100 scientists working on multiple iPSC type technologies, which is what Sernova is focusing on. So we see these as great partners for Sernova going forward.
Good. Okay. Another thing I was looking at when I was researching Sernova for the podcast is your approach to what you're stating is this total regenerative medicine solution. And so I'd like for you to talk to our listeners about what that approach is and exactly the why behind it.
Yeah, so for cell therapy approach, it isn't just taking cells and then pouring them into the bloodstream and thinking that they're going to function. There have been multiple failures with stem cells that have been placed into the bloodstream, and then you don't know where the cells went. So we believe there's 3 parts of the technology to be able to move this forward to treat millions of people with these diseases. And one of these is you want to know where the cells are. And so we developed this highly vascularized organ-like device that is placed under the skin. So that's part number 1, and we have proven that in our long-term clinical trial in patients that it's working. The second part of this are the cells. All right, so we started off working with human donor islets, but there's a limited supply of these cells. And so by getting a stem cell derived technology that produces the right amount of insulin and the other hormones, then that provides us the ability to treat all people with type 1 diabetes. And the third aspect of this, third-pronged approach, is how do you protect these cells from immune system attack? So there are 3 different approaches. One can gene edit the starting stem cells, and we believe this is a potential way of moving forward to make those cells blind to immune system attack. So that's one approach. The second approach is a simpler approach, which is more of a polymer type approach. And there you're not altering the stem cells that you're working with. And what Sernova is doing is that we're working with a technology called Conformal Coding, which is a technology that we have a worldwide exclusive license to from the University of Miami. And here what we're doing is coating the cells with a cross-link polymer coating. And it has been proven to allow normal flow of insulin and glucose back and forth across that membrane. And the membrane surrounds the cell almost like a shrink wrap, so it doesn't take up any room in the device, which is really important. And the third part of it is it's been proven to show that it can protect those cells from immune system attack. So what Sernova is doing right now is that we have an extensive collaboration with the University of Miami, and we're combining the technologies within our cell pouch to be able to drive this into clinic. And then that would eliminate the need for the anti-rejection medications that are used by all companies right now to protect those cells from immune system attack. The other approach is what's called tolerance. So you can also work with the immune system to teach the immune system how to tolerate the cells that you're putting into the body. And Sernova is actually working in the background on multiple types of approaches for protecting these cells from immune system attack. So just to summarize, again, there's three parts. There's the device, therapeutic cells, and then what we would call the local immune protection of those cells. And that will provide the fully functional product that we believe will be able to be used by all people that have these chronic diseases, irrespective of age over time, once we prove safety and efficacy of these long term.
Good, okay. I also want to talk, I want to hear from you, your perspective on what you feel based on your professional background and your work that you're doing with Sernova. What do you feel is truly the near-term future of regenerative medicine as a whole?
Well, I think what I see is this is going to revolutionize medicine. And what I think is important right now is you not only have to have the science going forward, but you also have to have the investor interest. And what we're finding is that because these studies cost quite a bit, and to get them proven, what we're finding is there's a significant amount of institutional investor interest in this field, and we're seeing that the investments are growing considerably. There are a number of cell therapy companies now that are moving forward, and I think we need to continue with the successes that we're having. So our success that we have, the data that we've come out with the American Diabetes Association meeting is a significant indicator that there is the potential to get this functional cure, and we just need to keep getting data coming out around the stem cell derived technologies, and we're not the only ones working in this field. And it just is, as long as we continue to get successes in the field, I believe very strongly that therapeutics is going to be moving from just masking symptoms of disease to actually be able to find a functional cure. And I think we're very close to that in multiple areas. And the first thing, we may have that functional cure, but we have to drive it through the clinical programs and, through work with FDA very closely to get these products approved and into patients as quickly as we can. You can imagine, even with this, the data that we've got now, we get multiple inquiries from parents who have children with type 1 diabetes. And it's really the patients that are driving this disruptive technology, and they are the ones that really need to keep pushing to get these products approved in a safe way as quick as possible. So
good. Well, and I was just about to say that there are listeners either professionally or personally, certainly deal with type 1 diabetes, and we all hope that this is curative much sooner than later. So wonderful. We've kind of reached the formal end of our discussion, but I wanted to ask you, when I was doing my research for today's podcast, your undergraduate and graduate degrees are in zoology, before moving on to earning your Ph.D. in pharmacology and therapeutics at the University of British Columbia. So what I wanted to ask you is, what led you to studying zoology, and how does that impact what you're doing today? How do the two meet up?
So I have been a biologist and interested in the natural world ever since I was a little child. And so when I did go to university, I became very, very interested in how different species in the world survive, extreme environments, right? How are their bodies adapted to do that? So I studied environmental physiology quite a bit. And after I did my initial graduate degree, I ended up working in the cardiology department in Vancouver, and really learned about the relationship of, human disease and how do we work to find cures to help these patients out. And so I kind of moved from animal environment physiology really to working in humans and how they adapt to disease and how we can then cure them, essentially. So that really built my interest. And when I did my PhD in the diabetes area, you really learn about how far behind we are in terms of coming up with a better treatment for type 1 diabetes. And, to think that patients have been on taking insulin injections for over 100 years with, what I would consider no really significant improvement really tells you essentially that, we can get stuck in the dogma of good enough when we need to get something that is an actual real cure. And so my entire life has been dedicated to disruptive type technologies that can make very significant changes in how people are being treated, and how do we help physicians look forward instead of accepting what's already, the good enough type approach. So it really, getting into human medicine just really expanded my excitement in being able to have a super contribution to the world in terms of helping to treat these diseases and changing the way people look at potential and look at great things that can be done in medicine. So, and the other important part of this is that it takes huge teams to be able to make this happen. It takes a lot of support, a lot of trust, and even, in companies that are competing with each other, we release data in medical conferences, and in many ways we're helping each other out to get to that functional cure for all people that have these diseases. And ultimately, it comes down to the people that we're helping, really. So, long-winded way of saying of how I got to this area, but I'm really thrilled that we're having an impact on people.
That's
and
wonderful.
their lives. So
yeah, absolutely. And I think that it wasn't long-winded at all. I think it was a really interesting path, and I love the approach of patient first, and however we collaborate, there's enough sun for everyone. So that's great. All right, listeners, that wraps up this episode of Cell and Gene, the podcast. Thanks to my guest, Sernova Corp's Philip Toleikis, for his time and for all the insight you've provided today. This was great, Philip. Thank you so much.
Thank you. Thank you very much.
Sure. Be sure to visit Cell and Gene to register for our newsletter and to read our timely content written and curated for professionals just like you. We'll talk to you soon.